So we're going to talk about the transportation methods through the cell membrane. And you said you understand the cell membrane pretty well, so it shouldn't be too bad. We have over here passive, and over here active. The reason I'm setting it up like this is because I want you to be able to recognize keywords that will help you when it comes to tests. That when you see a question, you know immediately what type of transport it's talking about.
So I just need 2 columns.
Yeah, so 2 columns. You got your passive and your active. Passive means no energy.
It required.
Yeah, no energy. Active needs energy.
And the way that he explained it that helped me the most was the upstream thing.
Yeah, so I explain it like this. So, if we have a hill,
Okay.
there's our hill, and I have a rock right here. If I push this rock, is that going to require any energy to go down that hill? No. It's going to go by itself, right?
Yeah.
If I were to go from here, have my rock, and I want to get it to the top of the hill, I'm going to have to push it all the way. It's going to take a lot of energy to bring it all the way back up, right? So this is the high end, this is the low end. So in passive transport, we say it goes from high to low.
Because it's the easier way down.
On the concentration gradient, yes, exactly. This little, this slope or difference between high and low is called the concentration gradient. So if you see that word, concentration gradient, it's actually 2 words. This is what they're talking about. They're talking about that difference between high to low. So passive, it's high to low. It requires no energy, just going from high amount of things to low amount. Did he talk to you about solvents and solutes? So high amount of solutes to low amount. Or we'll talk about another example in case where it's a solvent. But what direction do you think active goes?
Low to high.
Low to high, perfect. Low to high. Does this all make sense? Okay. All right. So
Excited for the turn.
perfect. Okay, so we're going to start in passive. So these are things that require no energy. The first one is called simple diffusion.
Oh, yeah. That's the one that he calls, it's something else in the book though, right?
I think it's the same in the book.
Okay, there's one of them that he calls it differently than the book.
Facilitate diffusion?
Oh, yeah.
What does he call facilitate diffusion? I think your book calls it facilitate.
He says the book calls it. I don't know. I wish I had my stupid. I forgot my notebook. He says facilitated transport.
Yeah, we'll call it facilitate diffusion.
Okay, he said he calls it something else in the book on it, but I couldn't remember what it was. So
Well, maybe he's talking because that one says facilitated transport. We're gonna call it facilitate diffusion.
Oh, okay.
But yeah, I don't know what he could be.
Yeah, it's okay. I'll look for it after.
Yeah. Okay.
How about?
So we'll start with simple diffusion.
Okay.
Here's our cell. Okay.
Uh-huh.
So here's the nucleus. It's just here's our cell with our cell membrane. We have a high amount of stuff out here, a low amount of stuff out here. In simple diffusion, just goes straight through the membrane. You don't need anything. It's as simple as it gets. Don't need anything else. It just goes straight through that membrane.
Mm-hmm.
So that's how I remember. It's as simple as it gets. It's just, and it just goes from high to low, just straight through the membrane. The reason it's able to do it is because this is small, uncharged particles. So examples of this would be oxygen or CO2. Small, uncharged particles are able to just go straight through the membrane. So that's simple diffusion. Now if I go facilitated diffusion, which is our next one, it's still going from high to low, but this time, it needs something. You know what a facilitator is?
Yeah,
Have you ever-
like in like meetings, facilitators like the leader of the group or like
Perfect, exactly. So it's getting something done, making sure that it's getting done. Yeah.
Is that the little like box thing?
Exactly, yeah. So we have,
Like an opener?
we call it like a protein gate or channel. These proteins in the cell membrane allow things to go through from high to low. So they still want to go from high to low, but they need a way to get there.
Okay.
And the reason being is these things are too big and they have a charge. They just, like glucose. Glucose is a big molecule. It can't just slide through the membrane by itself. It needs a place to go. So, did he talk to you about his dam example?
Yeah.
Okay, so I'm going to explain with the dam example.
So they have to poke the hole through it.
Exactly, because you have a high amount of water that does want to even out with the low amount of water and wants to go that way, but it can't go through. It can't just go straight through that dam. That dam is there blocking it. Now if I were to poke a hole, this water is then able to come out. It's the same idea. It needs a place to go. It doesn't require any energy. That protein doesn't need any energy because it's not doing anything. It's allowing a spot for it to flow through.
Okay.
Okay. All right. The last one we're going to talk about in passive actually is a pretty big topic. It's osmosis. Now, what's the difference in osmosis than these two?
I don't remember.
These deal with the solute, so these deal with like the stuff. Oxygen and glucose, just the stuff. Stuff that's dissolved.
Is that supposed to deal with proteins?
This deals with water.
Oh, water.
Or solvent.
Oh, okay.
Okay. So that's the big difference in our chart on this side. Okay. So osmosis.
How do the proteins get through?
Proteins we get through any of the transports other than osmosis, depending on what size protein it is and what its function.
Okay, so osmosis is just for water.
Yeah, it's just for water. So imagine we have a cup of water. Okay? That is our cup of water. And I'm going to say this is pure water. It's just, you can't make it any purer than what it is. And then I drop my cell into this water. Now, cells, they're made up of a lot of water. So let's make this cell 80% water. Now I make it 20% other stuff or solute.
Mm-hmm.
Just stuff dissolved in that water, things in there. Okay. When we talked about the dam, we talked about how the water likes to even itself out. It would want to be the same level. It just, it prefer being like this,
Mm-hmm.
straight across. It's the same concept here. It wants to be the same percentage. It wants to be at the same level. So, water will always go to where there's more solute. They even things out. They try to even things out. It'll always go to where there's more solute. So in this case, where is there more solute? Hint, I'll write this on the board for you.
In the water? Oh, there's 0 solute?
In the water, yeah.
Uh-huh.
And there's 20% solute in the cell.
So it'll try and go out into the water?
It wants to go to where there's more solute.
Oh, but there's no solute there.
Yeah.
Oh.
So where does it want to go?
Into the cell.
Perfect. It wants to go into the cell because it wants to even things out. And if I were to switch, let's say that's 70% and 30%. Now, where will the water want to go?
Into the water.
Yep, I want to leave the cell.
So it follows this, in osmosis, it follows the solute?
It always goes to where there's more solute. Yes, exactly.
Which is the opposite of solvent, right?
Yeah, the water is a solvent.
Okay, so it doesn't attract to the water, it attracts to the
It attracts to
non-water.
where there's more solute because it wants to even out those percentages. It
Okay,
wants
that
to
makes
make
sense.
the level as similar as possible. Now I'm going to talk about 3 different conditions underneath.
That actually makes sense just in general, like how water is. Like it doesn't stay straight up and down. Like it always will like flatten out into, you know?
Yeah, no, water likes to keep things level. It likes
Yeah.
to keep percentage. It likes to keep everything nice and neat. And so it'll try its best to do that. Okay. So,
Except for Nuremberg, when that's
actually-
what happens.
exactly. So, if the water's rushing out of the cell, like we have in this case, it's called a hypertonic solution. Now, I have silly little ways to remember these, so you gotta bear with me, but hopefully they'll help. So, when the water's rushing out of the cell shrivels all up. It's getting all small. And so it's getting really, really small and it'll look like something like all shriveled up. So it can eventually die. If the cell's placed in a hypertonic solution, if the level's not close enough to keep it alive, like it'll shrivel up and die.
Is that when he did like the rotten milk example?
Maybe.
Kind of, because like it's hypertonic when there's rotten milk because like, I don't know why, or something like that, because it can't stay alive. Like I don't know, maybe it was something else.
It may be. I know he does use rotten milk example, but
I don't remember. Okay, anyway.
yeah, it may be. So when the cell shrivels up, it dies. This is called a hypertonic solution. The way I remember that is it's getting little, and little kids are hyper. So
Oh, there you go.
hyper getting small. And you remember me talking to you about how science we need to know those key terms, those words? Hyper is one of them. Hyper means under, like, it means like giving what's the best way to describe it? The other one, it's the opposite of hypo. And hypo is like under the amount.
Well, like hyper is like over, right?
Yeah.
Because like you're over hyper, like when
It's-
I
yeah,
have
exactly.
ADHD, so that's how I get over hyperactive disorder.
And so it's over the amount of solute. It's really hard when it comes to osmosis to think about the key words and how.
Because it means getting small, you don't probably associate over with getting
Yeah,
small.
exactly. Because it's not dealing with the size of the cell; it's dealing with the solutes and the water.